I'm ready. Hi, my name is Linnea.
I'm Bezia.
And I'm Linnea.
And this is episode number 15 of the Lemma Journal Club.
What is the Lemma Journal Club?
The Lemma Journal Club is the show where we read every paper to tell if you need to read it. You probably do. We have a terrific paper for you today. We are talking about reconfigurable self-assembly through chiral control of interfacial tension. This is a paper by our PhD advisor, Zvonimir Dogic, and I think it's the best paper we've ever covered in this podcast. Neither of us picked this paper. This is a community vote. The community vote is mostly us and probably people from Zvonimir Dogic's lab. If you want to understand me and Linnea and our sense of scientific aesthetics, this is the paper. So I would genuinely encourage you to either pause the podcast and read the paper, or after the podcast, read the paper. It's not a long read. It's very well written. And it really, I think, explains the soul of me and Linnea scientifically.
Well, so it's an interesting take that you think this explains the soul, because I don't disagree, but I had never previously read this paper in full. And Bez is leaning away from his microphone.
I made the mistake of taking a bite out of a cookie in the middle. I don't know what I was thinking. I don't know why I would reach for a cookie while we are recording. Jesus.
And I joined Zvonimir's lab for vastly different reasons than this paper. But it does reflect a lot of the underlying beauty in science that initially got me into it. And the work that I did as an undergrad is very reminiscent of this work. And so, yeah, I found it a weird resonance with Zvonimir's work, because I was aware of the work and I had seen the images, but I had never read the paper or seen some of the more detailed stuff.
I also did similar work to this in college, and I saw Zvonimir talk in my last year of college. And that is largely why I applied to work with him, because I saw him present this work and it was just the greatest thing I'd ever seen. And every once in a while, I take breaks from looking at this kind of work and I kind of forget what it's like. And then I go back and read one of these papers and I'm like, oh my God, I really remember why I was so fascinated with this. I remember coming to Harvard and people in the physics department saying, oh, you know a lot about biology, you study biophysics. And in my mind, I'm thinking that I'm studying these colloidal systems. And I was like, what are you talking about? This isn't biology. But it is, this is a podcast where we're talking about biophysics papers, right? It is worth saying that to physicists, if you're doing cosmology, this is biology. To biologists, if you're studying a mouse, this is pure and fundamental physics. And so this field is really deeply underserved field, because so many people scientifically just cut this out of what they think their broader field is. The reason why physicists think that this is a biology thing is because it's going through a material science where the material is made out of these programmed artificial structures that just happen to be viruses. The virus is the structure. They kind of look like a Twizzler, and we're building materials out of Twizzlers. But because it happens to be built out of a virus, this is a biology paper to many physicists. I couldn't convince anyone in Harvard physics otherwise. It was really, to me, the death of my love of physics departments was seeing how many physicists were just blind to physics when it no longer took the form they wanted it to. To biologists, this is a physics paper because it's completely artificial and it's about the structure of liquid crystals. And the biologist says, well, what could this possibly have to do with biology?
I think it has really interesting implications in both fields. But biology, there's so much coming at you. There's so much information. Maybe this is just because I'm not a trained biologist and I've been plopped down. And it's like, if I just call everything biology, I'm never going to feel like an expert in biology. And so you have to draw the line somewhere.
Yeah. This is a very dense paper. This is a lot of I think the purest form of a lot of Zvonimir's basic intellectual motifs put down in this paper, just sentence after sentence. You saw my highlighting of this paper. Every other sentence is highlighted, but it's also a very beautiful paper.
And it's almost, it's 2012. It's now 2026. So it's almost 15 years old.
Yeah. So I want to talk a little bit about where this paper went. So this paper, again, was what I thought was some of the most beautiful stuff. It had been going on for a long time with Seth Freyden and Zvonimir Dogic. You can
and
find
Bob Meyer is on this
Meyer,
paper. Yeah.
Rudolf Oldenbourg is on this paper. But the point is they had been doing this work for a long time before this. But this is kind of the magnus opus. This is the work.
I mean, this starfish, chiral starfish, which is the figure that I always reference, was the Brandeis-Mercek logo for most of our tenure at Brandeis.
So given that this is the work, it's a fairly old work, it's 2012, right? How many citations do you think it has?
So I know because I have it up, but it has 231 citations.
It is fairly down there in terms of citations of papers. In the same year, Zvonimir published Tim Sanchez's active matter paper, right? This was the beginning of Zvonimir Dogic's active matter push. This is what me and you did our PhDs in. I came in to Zvonimir's lab to work on these Twizzler viruses. I said, I'm really interested in these Twizzler viruses. I have all these ideas. And he said, I'm kind of transitioning away from that work. I just hired my last PhD student who's going to work on that. And I went off and did active matter. And that has, almost 2,000 citations, right? Almost an order of magnitude more citations than this reconfigurable self-assembly through these Twizzler viruses. It didn't get heavily acquired. It didn't create a whole huge field of its own like the active matter stuff has. And I know that's because it had been going on for much, much longer. But if you look at the citations of this paper this year or last year or 3 years ago, this is not like a beautiful burgeoning field that lots of people have taken off with and run with. There's a couple people who are kind of carrying the torch just a little bit of like how you could use this to fabricate interesting patterns in material science for microfabrication, which is where it belongs. But, this didn't take off. And really the person who carried the torch was Joya Miller. Joya Miller, during our time, put out this unbelievable PNAS paper that I went through the PNAS paper again to see, do I think the PNAS paper is really the magnus opus, or is it really this nature letter? I do think it's this nature letter by Thomas Gibaud, who, do you know?
Yeah, I was going to bring that up that I don't know.
I know Ed Barry.
Yeah, I know Ed Barry. I've heard of the middle authors, Mark Zakhary, Andrew Ward. These are all names I saw around the lab. But Thomas Gibaud, he must have been a postdoc or something in the lab because I never saw his name around.
Thomas Gibaud, if you're listening and you're still in science, send us an email because we don't know who you are. And we're like intellectually related to you.
But yeah, so I mean, like this field, like it is really just, it's, I think, a testament to how one person's fascination can just drive really beautiful science. And even though, it hasn't started a whole field behind lots of lots of people joining into this, I think it's inspiring work.
But why didn't it? I think it didn't.
I think it's really hard work. Do you remember in the Dogic lab?
I understand that experimentally.
No, yeah.
My question is, theoretically, why do people like Cristina Berciu want to do active matter physics rather than this chiral fluidity physics? That's my question. It's not why there's not that many, because there's not that many active matter experimental. Some experimental systems are hard. Fine. I buy that. My question, from a theory perspective, I kind of think everything is equally hard. Why didn't the theorists jump on?
I think it's in some ways known physics.
You think it's hashed out?
What they're doing is they're getting to very low surface tensions. Like it all fits into your normal framework for interfaces. And so there's no, like when Cristina Berciu is writing down a new active matter equation, it's like, what are the terms? And they're taking guesses at what they are, and then they're seeing what comes out. And that feels like a very different endeavor than here, where it's like, okay, we have this chiral term and it so clearly captures the data. So there's less mystery involved on the theory side.
Yeah, there is. I think that's why I love it so much, is because there's less mystery. You can really use physics as a tool a lot more.
Yeah.
And this creates these beautiful things like chiral self-assembly produces structures that combine seemingly divergent properties of high elasticity and fluidity is one sentence that I love.
Yeah.
And the sentence that I really love is in the beginning, this kind of bold abstract. An example of particular interest is smectic liquid crystals, where the two-dimensional layer geometry cannot support twist, and chirality is consequentially expelled to the edges in a manner analogous to the expulsion of a magnetic field from superconductors. I love this idea that the chirality is expelled to the edges. Do you want to talk about what liquid crystals are?
Yeah, so this actually gets into one of my questions about the paper. So liquid crystals are materials made up of molecules that have some aspect ratio, some long aspect ratio, so little rods that tend to align with each other, but still form a liquid. So there's no positional order. Yeah, I mean, one question I had here, so these FD viruses that they use in this situation, I think it was known previously that at high densities, they would form a liquid crystal.
Yeah.
Yeah. And so was this Zvonimir's innovation, or this was Seth Freyden back in the day, was to put them at low density and add in dextrin to get these membranes to form?
That's a good question. I know that there are papers with Seth Freyden as the last author on them in which you have these chiral viruses making twisted ribbons. They hadn't done this pole scope stuff. It doesn't look that amazing. But I do know that there is at least one paper with the twisted ribbon where Seth Freyden is the last author, significantly before my reading time. It might be like 99 or something crazy.
Yeah, and there was also these mutant viruses that have no intrinsic chirality, or like no ability to tune the intrinsic chirality.
Yeah.
And who made those? So I had just some questions about the history of the field.
I think the building blocks are all Seth Freyden.
He was just became enamored with how viruses, how
it's a good
uniform
question.
viruses are
actually
for
looking at
colloidal.
it, I guess I know why. Seth Freyden is not on this paper. Isn't that fascinating?
Yeah, I mean, it must have been for Zvonimir's tenure.
Yeah. That they made a decision. Yeah. But it is an interesting question, right? The torch was carried by Zvonimir, and Zvonimir's prose in writing it is so deep because even as an undergrad, he was working on these processes. So there's like some really deep intellectual thought that just grips me. It's really Seth's thing, and maybe he got it from somebody else, but like the furthest back that we know the lineage is from Seth, and we've never talked to him about why he got interested in this kind of work.
Yeah, next time we see him at a conference.
Yeah,
so I think what's happening is, that there's a liquid crystal tends to be a three-dimensional liquid with all aligned rod-like molecules. Yeah, and what they do here is they reduce the density of the rod-like molecules and add in a depletant. And this allows them to form these single-layer aligned liquids.
These isolated structures. And when you say single layer, are these on an oil-water interface? Or are they just sedimented down?
I think they're just sedimented down because they say it's polyacrylamide. So they don't ever talk about the oil water. But they are like fluctuating around a lot. So
they do say fairly early on in the paper. So now I'm going to start asking some questions about the paper. They say, despite having different structures on molecular length scales, the coarse-grained properties of colloidal membranes are identical to those of conventional lipid bilayers. However, unlike their amphiphilic counterparts, colloidal membranes do not form vesicles and are instead observed as freely suspended discs with exposed edges. Mm-hmm. Okay, so now in the 10 years, 14 years after this paper, I know that there are virus papers written by Zvonimir and co. about the viruses forming vesicles. What's the trick there? Do you remember?
I don't remember, but I think you have to make a really, really big membrane. And then I think you have to use a specific composition of viruses to make the edge particularly unstable or something so that it'll curl up. So the energetically favorable state is this.
To not have an edge.
But yeah, that's a paper I would love to do for this podcast as well. I'm kind of waiting for the holography paper on that one.
Oh, that would be a great one.
Yeah.
Yeah, if the holography paper comes out, that's the one to do.
Yeah.
Absolutely. Okay.
Conoscopic. Had you ever heard of this word before?
Where is it being used?
All the time.
Is this like the pole scope stuff?
The micro lens pole scope. I mean, you can figure 1G,
This
conoscopic image. Like, I couldn't figure out, I tried to go back to the original. So this is from a Rudolf Oldenbourg paper, unsurprisingly, this 3D polescope technique. And I cannot figure out whether, like, why they call it 3D. And so do you remember Joya doing tilted polescope?
Yeah, but that's not what this is.
Okay, but I feel like it's giving you similar information that they say in 1E, like, look, we can tell that it's tilted, right? Because of the bright edges.
Yeah.
And so this is because those bright edges are causing the light to move differently through the membrane.
Yeah.
And then they're like, but we want to know which way it's tilted. And so they do this 3D polescope where they put a micro lens array on the image plane.
Yeah.
And then this breaks up the signal into like discrete boxes.
Yeah.
And so you can see in each box where the viruses are, like, which direction they're tilted towards.
Yeah, and each box is called a conoscopic image.
And so I think from brief Googling, I didn't get a chance to read in depth, but conoscopic is just any like lens, any image created by a lens.
That's not useful, but okay. Yeah.
Make observations of a transparent specimen in a cone of converging rays of light. Here we can tell which way the rods are tilted, right?
Yeah,
in
that's
G
the
and
idea.
H.
Yeah.
Okay, but what was Joya's thing then? I thought tilted polescope was supposed to resolve this inability to, like, I thought that all polescope was...
We didn't have a 3D liquid crystal polescope in the Dogic lab. We didn't have a micro lens array. And I think they must have not liked this strategy in the long term because they didn't continue to go and do this in Rudolf's lab. Like, when we visited Rudolf about 10 years ago, he showed us a camera, a physical camera, and he took our picture.
Yeah.
Just a picture of us, not a microscopic picture. And the camera had these conoscopic micro lens array, right? And so it took a 3D picture of us. So he was still really interested in this stuff, but I don't think that this became, like, maybe there's a thing where there's a little bit of the measurement that they don't quite believe, or it's really a two-dimensional measurement, but there's something about the data that they don't like. And so they didn't go on to produce it. So we didn't have it in our lab. That's my theory. You want me to call Joya and ask her why she used it?
Don't call Joya. She's probably at work.
Hi, Joya. Do you have a second?
Yeah.
Lainey and I have a Dogic Lab question for you.
Okay, let's see if I remember anything.
In Zvonimir's 2012 Nature paper on the virus stuff, they use a 3D liquid crystal polescope, which is Rudolf Oldenbourg's like array of little conical things. But then you, in your papers, just take the whole top of the microscope and tilt it to the side. We think to get similar data.
Pretty much.
Why did you not do the 3D liquid crystal polescope thing?
I don't really know. It was like it wasn't an option.
Because we just didn't have one.
Like, dude, we didn't have one. I'm not sure if Rudolf Oldenbourg even had his was accessible. Like, yeah.
That his maybe didn't exist anymore.
Yeah. Or like wasn't, yeah, pretty much.
But it does, in the end, you decided that it gave you very similar information to tilt the bright field arm.
If you basically, you didn't really tilt the bright field arm as much as you moved the main light source off center.
Is that not the same thing?
It is the same thing, yeah.
Okay, great. Thank you so much. We might call you again with another question or two.
Okay, cool.
Okay, bye. Sorry to bother you.
No worries.
Okay, that's what I thought. They didn't have it anymore. This is the thing that bothers me about this whole paper is this stuff it didn't flourish. It didn't get adopted. There isn't a whole community of people studying it. I thought this was the coolest stuff in the world, and it didn't explode.
I don't know. This is one of the things that actually drew me to soft matter initially, was that you could be a single person with a single brain and a single set of skills and like solve problems that were beautiful. And this does not lead to whole fields of research, but it does that this is one of the beautiful things about soft matter to me. Do you remember in the Dogic lab, there was a bottle of dextrin that was like,
don't touch this
don't
dextrin.
touch this dextrin. This is the best dextrin and the only dextrin that can ever be used.
Yeah.
So I think that the chemistry and the of these bio-inspired materials can be really, really finicky.
I will say, and this is a very physics thing that doesn't exist in biology at all, and kind of seeing it here kind of shocked me. The 3D images that they create, which are just, someone going in Blender or whatever and making this beautiful rendition of what they think it looks like, do not look at all like the data that they have, right? The data that they have is bright and dark regions, and they interpolate through a bunch of physics and a bunch of creative thinking. Oh, it must, at the nanoscale, look like this.
Yeah.
Totally doesn't exist in biology. To take these images in like D, G, H of figure 1 and draw figure 1b, to take the images of figure 2f and draw figure 2e.
Well, it's very different, like just very different interests, right? Like biologists will draw these gene cascades or like,
those are terrible.
like this transcription factor activates this thing. And then so they draw, but it's not a physical picture, right? It's this like chemical signaling picture.
If you could get rid of one of two things in biology, either the gene cascade diagrams that have no like cemented nomenclature, or these principal axes things for genetics, where they've just created two completely useless artificial dimensions, which would you get rid of?
The gene cascade diagrams.
Yeah.
I feel that I know the principal axis means nothing, but how else are you supposed to look at this data? Like, I understand that there's a limitation there, and I think it can be useful to just like try and separate things out. I don't think that these like arbitrary networks are very helpful.
But I was like, I really would like a super well done cryo-EM of these virus rafts. I don't know if it's possible to do. Does it really look like that?
Okay, yeah, I was super disappointed by 1D and the supplementary figure, which I went to go look at because I was like, ooh, like that's cool that they've actually seen the individual viruses. And like, I remember people showing the like one picture that everybody showed of a single FD virus, which is this like long, skinny thing that like looks like a rod. Kind of crooked, and I was like, "Ooh, it would be beautiful to see the little, like the real Twizzlers, like you're talking about." And it's so disappointing. Did you look at the supplementary figure?
I looked at 1D, and that's what made me make this comment of like, man, they've just like drawn a thing that they have no direct imaging evidence that it looks like that.
I mean, I like believe that it's evidence that it looks like this.
Sure, in the physics sense. Not in the biology sense.
They do, I think they even do cryo-ET. They do some sort of three-dimensional cryo to try and get a better sense in the supplementary figure.
Oh, but look at this. This is ReadCube, again, is doing the supplements, right? It's doing the movies in, right? I can look at it in line, right? Supplementary figure 3. Oh, yeah, this is useless. Well, this is better than what they have in the main figure.
Sure, it's better, but it's like I was looking for something that really supports or like really like evokes C and B, 1C and 1B. But it's these viruses, and they say this in the caption that they've shortened the viruses for display. And I remember the virus people in the Dogic lab being like, if you really were to show the whole virus, it would just be like, it's so tall. The aspect ratio is so long.
Oh, I wish they had. I guess it doesn't look like this. I really wish they had like a real image. I know it's hard to show graphically, but to get closer to the real dimensions would be really helpful.
I think, I mean, sometimes in their presentations, they would do this of like, this is what it would be like. And then here we're shrinking it. This is what we're saying. This is and so you could get a sense, but you really can't display it in this with their real aspect ratios.
Here's a fun complaint about this paper. Some of the figure captions are not sentences.
Yeah.
I like that complaint because who did we learn this rule from?
Do you really think we learned it from Zvonimir?
How could we have not learned it from Zvonimir? How could it come from someone else?
I don't know.
Did it come from Dan, is what you're suggesting?
I'm suggesting I learned it in my postdoc, yeah.
Oh, I definitely didn't learn it in my postdoc. I definitely learned it in my PhD.
I guess I should go back to my old papers and see if it's in there.
Oh no, you're suggesting that like my PhD papers don't have this, like my first PhD paper doesn't follow this
I'm
rule?
suggesting that for myself, yeah.
Oh wow, interesting. Okay.
But obviously didn't bother me here because I know the project so well, but I stand by the sentences as it just makes it so much more digestible.
Getting a little bit into the physics, figure 2D.
Yeah.
Did you understand what figure 2D was supposed to be? Is it at a given temperature? They say that, yeah, they go through many dextran concentrations and they say those fall on a line. But I thought to change the chirality, they have to change the temperature. Can I think about the y-axis as temperature? Like that's what actually is happening?
I think that's right, that the y-axis is temperature.
Yeah, okay.
And the x-axis is the wave number of the fluctuation.
Okay. Right. Actually, the first time that I read through it, I wasn't able to figure that out. It's only...
Is that true, though? Because the 2C...
So 2C shows you the effective line tension versus temperature.
Yeah.
From the effective line tension, you can back out what the chiral line tension component is and what the non-chiral tension
So I
component
don't
is.
think that the y-axis then is at all related to temperature. I think what they're saying is that it's independent of...
No, the chiral line tension is definitely a function of temperature. They show that in the text.
Yeah.
So I think that I didn't capture this interpretation correctly the first time, but I think the correct interpretation of 2D is, as we said, the y-axis is essentially temperature that they've converted to physical units of the line tension of the chirality component, and they plot that against this wave vector.
And so if you were to make if you were to plot chiral line tension versus temperature, it would just be a flat line.
Chiral line tension versus temperature.
So like make the C plot, but for chiral line tension.
I think it would look identical.
So it would correlate. So what C tells us is that the effective line tension increases linearly with temperature.
Yeah, so the effective line tension is going to be the chiral line tension added to the non-chiral line tension. There's a sign thing there.
Yeah.
But basically, if you were to plot the chiral line tension here, it would just offset 2C, would just offset this graph by a constant because the non-chiral line tension is not temperature dependent and therefore is a constant.
Okay.
So it would look exactly the same, but shifted.
Then I stand by that the y-axis in D is not just temperature.
No, I think it is, right? Because it's temperature times a constant.
Correlates with, it's linearly
correlated
with temperature.
And therefore, I can think about it as a temperature.
Okay, sorry.
It doesn't have units of temperature, but I could say the thing that is changing in the y-axis experimentally is temperature. Okay. That's the main thing that I want to say. Yeah.
Yeah, I think that's what C is showing you. C is showing you that as you change temperature, you're changing the effective.
Yeah.
And so C allows you to make that D.
Yeah.
Okay. Yeah. Yeah. So this is.
So I guess we should talk about the trick that they're doing a little bit, which is essentially they can get their Twizzlers to straighten out and become normal rods or to become more Twizzlers by changing the temperature. And that allows them to dynamically change the chirality of these virus materials, which makes the materials undergo crazy shape changes that I cannot even describe. You must look at some of the videos we'll upload or the pictures from the paper. It is geometries, and some might argue topologies. It's geometries that are completely different from what you've ever seen. It's like a disk turns into a many-pointed star, except the points of the pointed star are twisting around as they go outward. And then you can take some of these bizarre shapes, you can take the ends, you can use these optical traps to combine the ends and create new topology shapes that are genuinely new topology now, which again are not shapes that you encounter in life and thus don't have words to describe. They're
Okay, you say don't encounter in life. So we don't encounter them in our everyday life.
Sure.
My question is, are these shapes in biology?
4B, this Twizzler, where it's been wrapped in many, many, many circles, and then the edges glued back together. I have difficulty believing that shape, well, I don't know. That might be what E. coli RNA looks like now that I think about it.
I'm not saying that all of this exists.
Yeah.
But I think, one of the really like what actually brought me back to this paper was thinking about a biological system and what could possibly be happening there.
Yeah.
And how do you get an intuition for it?
Yeah.
And going back to this paper, it was like, oh yeah, wild things can happen because a membrane in biology can have chiral components, right? Like that can be a part of a membrane.
Yeah.
And so you can undergo really unintuitive shape transitions.
Yeah.
We don't think about that when we're thinking about art. I mean, I don't think about that when I'm thinking about biological membranes typically. And from talking to some people in the membrane, or at least adjacent to the membrane field, they're not thinking about these like what's changing about the physics and what shape transitions can you get. And so I would love to look, just go back through old TEM images and see if there's any hints of...
But you're not going to see it in TEM. That's the thing, right? Just like we don't see it here in their cryo, right? Like, I think you really got to find it in bright field is part of the trick.
Oh, so just like look with Polscope.
Yeah.
Yeah.
And so then there's two questions. One, can you find it happening naturally in nature? Is there naturally in nature? Is it terrible? Can you find it happening in nature? Can you see like a beautiful Twizzler star somewhere in nature and be like, oh, that's a Twizzler star. That's the shape. The other thing is, can I take like a mitochondria and force it to do this and insert into its inner or outer membrane a chiral component and force it to do this? Yeah, exactly. Yeah. Can I? And is there any organelle or any cell or any bit of biology where you can force it to do this in the biology and see how it reacts? Yeah. And it's like, would that be useful? Like, how does that improve? Like, is there any evolutionary pressure for this to happen? Or like, yeah. But this is why I talk about aesthetics, is that like none of this is, I think, the most useful thing to possibly be looking for in biology. But it is maybe the most beautiful thing to us. And I think that was a huge, you talked about this once, or maybe not. There was a sense in the Dogic lab of shared aesthetics. Yeah. Of everybody was interested in the same sort of scientific beauty. And that really pulled the lab together so that everyone would row together, is that we were an incredibly diverse lab in a lot of terms, but particularly in terms of intellectual thoughts, that a lot of us disagreed on basic things such as good and evil. Like there were some really large discrepancies between what people believed in that lab. And we all loved each other. It's a great thing to remember for lab management as we are trying to transition to more lab manager roles, is like this sense really, of shared beauty. Getting everybody into the same science boat really makes a big difference. One thing I was thinking about while reading this paper is, can you make this system a little bit bigger? That is rather... yes. Length scale is an interesting thing here and something I don't have a good grasp on. Well, I want the constituent particles, the constituent colloids, to be larger than a micron. That is their smallest dimension should be larger than a micron. Okay. So the radii of the Twizzler, I would like to be larger than a micron. So I think this is, is that possible? People are doing, Seth had moved on to some DNA origami stuff. I forget how big those are, but they're definitely bigger. Maybe that's what the flagella are. Flagella, their largest is definitely not a micron. I went to Nick's talk recently. Their radii is on their shortest axis is still smaller than a micron? I would say so. It was diffraction limited, it looked like. Okay. But I really need larger than a micron on the smallest axis. And the reason why... So I think what they're doing with the DNA origami is like... Right. The goal, I think that they were proposing, or one of the goals, was to be able to make any sort of geometry for colloidal. So you could, even if each individual one, you could get them to mega assemble into subunits that were of whatever size you wanted. So something I think about all the time for colloidal experimental control is Emily Gerrels from Vinnie Menaharen's lab. She now runs a lab doing morphogenesis stuff somewhere in France. For her PhD, she had a system in which she had colloids that she had colored red and blue. And she could shine red light on the red colloids and increase their temperature without increasing the blue colloids' temperature. Mm-hmm. She could shine blue light on the blue colloids and increase their temperature without increasing the red colloids. And thus, she was able to spatially pattern, to some extent, not exactly, but spatially pattern temperature right? And you could imagine going further with this, with really doing like a programmed light where you only increase the temperature of this patch of blue colloids. I thought it was one of the best experimental ideas to study entropy that I had ever heard, right? I really was blown away. Again, it got no traction, and I'm just like confused on what people are doing or what they think they're doing. But I would love the ability to study a system like this, where the chirality is tuned by temperature, where these things are large enough that I could make one blue or I could make one red because they're now larger than light, and I can start to color them. And then to tune subpopulations of temperature is something I would really like to do. But in order to do that, you need these much bigger. And I don't know if the physics works, because, you know, once you get up to 2, 3, 4 microns, your Brownian motion starts to become quite subdued, right? And I don't know if you get to create anything like this.
Yeah. And like also, can you undergo these chiral transitions when you're that much bigger? Like you might need much larger changes in temperature.
Yeah.
But yeah, the question that I find, or like that I wanted to talk about with length scale is how does the length scale compare to a biological membrane? In terms of the individual molecules. And I don't
like
have
figure
a good sense.
3A, for instance, all the scale bars are supposed to be 2 microns. So 3F looks pretty biological scale to me. 3A, maybe a little bit less, but yeah, it seems like it's...
Yeah, I just, like each individual, do you know how long a fat molecule is?
How long a fat molecule is? No.
Like the thickness of the membrane compared to the size of the structure, essentially.
Yeah, okay, I hear you. I understand what your question is. I don't know. I don't even understand really how the thickness of these viruses. Do you know the thickness of the viruses? How tall is an individual virus?
I thought it was like a micron or...
Because you were saying that these figures are highly deceiving, that they're like, you know, artistic sketches.
Yeah.
So no, I don't know
I'm like back through my brain of many Dogic lab, but I forget what the scale bar is. I thought it was like a micron.
The longest length scale of any of these things in both like lipids and in carbohydrates.
And I do think that it matters, right? They would make like longer viruses and shorter viruses, and this is how they could get these like weird rafts inside.
Yeah.
And so I do think the aspect ratio and the length scale matters here, and to have a good intuition. Our chickens are upset about something.
I was gonna go check on. Okay, extremely brief intermission to check on what the chickens were complaining about. They're panicked about something, but they've all gone back inside, so I think they're going to be fine.
We'll try out some new intermission music. Yeah, okay, and so, I mean, this figure 4 reminded me of writing papers with Zvonimir, or like dreaming about papers with Zvonimir. He really wrote short papers, or encouraged short papers. I know your paper with him was...
Have you seen our PRX
But that
paper?
was, I would say, a Dan Needleman decision. And where it was like, okay, figure 1, figure 2, figure 3, what should we do for figure 4?
Yeah, figure 4 was the dream figure.
Yeah, figure 4 is the figure like where you're not really going to explain any of the physics. You're just inspiring future work and future investigations. And yeah, so this figure 4 of like joining the ends of the membrane to create this like Mobius strip. And like just showing pictures of these super coiled membranes, doubly twisted, I think is what they called them, really reminded me of, just like have a dream, like do a lot of physics, show it really deeply, and then put a figure at the end that's like, okay, where is this work going next? What are the things that it can do and that this work can explain?
But we touched on this a little bit about why the theorists are drawn to active matter and not to this. It is one thing that I was really, sad about during my PhD is how little physics could really do for me. That it felt like physics was a cherry on top of an ice cream sundae that had been entirely been made out of material science and biology. Here, it feels like physics is the ice cream sundae. The biology is the cherry on top. And it's because you're able to use things like line tension and surface tension and chirality to really predict and understand the system, which you really couldn't do in active matter. In active matter, you were always trying to shoehorn physics in, but it wasn't like super predictive or super powerful. And I've just been sad about this ever since. That my whole PhD, I was sad about how little I was able to really do deep predictive physics. And that's continued on during developmental biology, of how little I can use deep predictive physics. Most of the physics that I'm using is explanatory at best.
Well, I have two things. Hopefully I can remember both of them. One is, do you think that they thought that this is what they would see?
Yes. I think that Zvonimir imagined this, or maybe Seth, that somebody imagined this in their mind in a similar way to I think they really did understand how to make the active matter system, even though they didn't know exactly what it would look like when it formed.
Yeah, I can see that they thought of this competition between the chiral component and the regular component. Sorry.
Yeah, the packing.
And that might lead to some shape transition, but whether this specific one and that this theory is what they had initially envisioned. Like, I guess I'm wondering, like, was this a theory-inspired experiment or an experiment-inspired theory?
That's why I say that this is so good, is that to me, this dissolves this bullshit arbitrary theory experiment border. This was physics.
Yeah.
They knew the physics and they used the physics and they learned that the physics works. And this is a great triumph. And they built a system that you can now explore more physics off of, right? That this is real. And that everything
But then
else
I don't understand.
Always has a little ring of fake behind it.
Because you say explore more physics. Like, you don't need to use experiments to explore physics. You now have the equations.
No, because math always works, right? So when you've got equations and you've got math, you can always explore the ways that math works. What you don't know if this corresponds to a physical reality. Just because it's a mathematical reality doesn't mean it is or is not a physical reality, right? And so physics, as opposed to mathematics, is exploring what we think and replacing it with what we know. And here you're really doing it. You've got a big book of what we think and what's possible. You're exploring new things and finding out what we know, right? And you're building up off of that. And sometimes that creates new little pockets of mathematics that you hadn't explored before.
Yeah.
Sometimes you find, I can build this all off of mathematics I already had. And some people feel that one of these is more important than the other. It's all bullshit, right? But this, to me, was the beauty. Like, I did this in my undergrad, and I did these huge, wild equations, and I said, okay, these equations are nice, but do they mean anything? And my undergrad advisor said to me, what do you mean, do they mean anything? If this isn't true, then physics doesn't work. This better be right, right? And then we did the experiments. It was right. It was like this holy shit moment of like, oh, we're not just playing around here. We really understand some bit of reality.
Yeah. So this is the second thing that I wanted to mention, which is I've been talking to you a lot lately about what am I doing in the lab? Why am I doing it? And I think this is one of the things that excites me about this in vitro project that I'm working on, is that it really feels like if it works, like we're exploring physics in this same way of like, okay, like we can see what's happening experimentally and we can write down the physics, and it's not a area that has been observed before.
Yeah. And this is what I'm trying to bring into developmental biology by considering energy, but I'm having trouble getting people to give me a leash to really quantify energy in joules so that I can move away from like descriptive stuff and move towards this much more solid, this is what reality is stuff. But yeah, I mean, just the fact that we're talking about such a heady piece of intellectual stuff through this paper, again, it's this beauty of doing things in this manner. If you could...
Sorry, before we get on to I assume you're asking the end questions.
Yeah.
Before we get on to the end questions, I wanted to say one thing about the legs of this paper.
The lack of legs.
What you say are the lack of legs, yeah. So it has very few citations, but I do see this work brought up a lot in talks. And when people are talking about liquid-liquid phase separation, which is a field that I'm in right now, weirdly, this work comes up.
Yeah, I can see why,
Yeah, and it's like the depletion interaction and also just like, how you can get particles to separate in a liquid from the bulk.
Depletion interaction, by the way, one of the worst named things in all of soft matter physics. I remember saying depletion interaction as an undergrad to a biologist. And they were like, what you mean is like running out of something? I was like, oh, that is what that word should mean. And they're like, that is what that word means in my field. Crowding effect, I think, is what soft matter should be using.
Okay, yeah, that's an argument for another time. I haven't given it a lot of thought.
Yeah, I see why it's so popular. Maybe, again, it's like,
They don't
rail
show
all the time
they don't show
against-
these beautiful phase transitions, but they do show like the membranes themselves or these Joya type things.
No, maybe you're right. Maybe it does have legs, because I do rail all the time that the number of citations of a paper is not remotely a good metric for how good the paper is, how interesting the paper is, how many people are interested in the paper.
Yeah.
Right? It's, again, I have the ability to get a number, therefore I feel compelled to use that number, but it's a bullshit number. I really hate that Google Scholar, which is what everybody is using, gives you two pieces of information. They give you the number of citations of a piece of paper, and they give you the H index of the author. And it's like, man, you don't need to report either of those things to me. You are shaping what the field cares about because you've just programmed in these two things to your stupid Google Scholar page. So you're right. If everyone's showing it in presentations, it doesn't really matter that people aren't citing it. It still has legs. I'll agree with you on that. And it certainly has, it's stuck around in my mind, that's for sure. If you could add anything you wanted, one experiment to this paper, what experiment would it be?
It's an interesting question because so much, Zvonimir has done so many more experiments with this system. And so, it's a hard one for me to answer. I guess I'm going to kind of take a side route here, back to talking about my desire for confocal pole scope. That everyone keeps saying 3D pole scope, 3D pole scope, 3D pole scope. I want confocal 3D pole scope so that you can look for these transitions in biology.
So what you want here, in biology, not in this system necessarily.
Not in this system, yeah. So like going back to our conversation about like, does this exist in biology? To me, that's an exciting, beautiful question to pursue, but very technically challenging because pole scope, right, this is a 2D system that they're looking at. And pole scope is, as far as I understand it, an inherently 2D technique. So how you go through a whole cell and find...
So were you not happy with episode 5's polarized fluorescence light sheet microscope?
Yeah, so we could put in fluorescent reporters on a membrane protein.
Yeah, I guess this does bring it back to, though, we want to see I just fully looked completely away from the microphone while speaking because I was like daydreaming off into the side. I wonder if
you got to daydream into the microphone.
Yeah. I guess, again, talking about the problem with the fluorescence microscope is it's only reporting the one thing that you labeled. So if we're looking at like a mitochondria membrane, we're only learning information about that one protein that we labeled, and we might miss something critical about something else in the membrane.
Yeah, and this concept that you brought up yesterday or something that one of the biggest opportunities in biology is all of the dark biology is what I like to think of it. All the stuff we don't know yet. One of the other Hannah Gray fellows does lipid biology. And so usually in biology, we think of all lipids. And when we talk about it in this podcast, right, we think about all lipids as the same, but every membrane has different lipids in it. And so even if you were to just label the most common lipid in a cell, you're still biasing your measurements.
But also I think like labeling a lipid is super, super tricky, right? You can label it with a dye, but I don't know if you can label it with like a genetically encoded fluorophore in any good way. And then even if you do, either way, you've changed the structure of the lipid by labeling it in a probably disastrous way. I tried to do some lipid labeling in some tissue systems, and it seemed to significantly change the biology that was going on by labeling the lipids. I don't know the answer to my own question. I don't know what experiment I would add because, again, so much has been done. One of the other coolest things that I think happened was these raft-raft interactions. That's Joya's paper. Again, I think for me, if I could do anything, it would be a mesoscopic analog to this experimental system. I don't know what that would be, if that's physically possible. I suspect it's not. But if one could take a picture of an individual rod through light microscopy such that you resolve the thinnest dimension, I think that would open a whole new world to me of things that you could explore. And that's what I would want to do. Another way of saying that, even larger, I think what I really wanted is at the mesoscopic scale, but even larger at the macroscopic scale. Can you create a system which expels its twist to the edges because it doesn't like that the internal particle is chiral? To see that macroscopically would also be super cool.
Just a detailed question of this paper. there was twist at the edges even when the molecule wasn't a chiral.
Yes.
Yeah.
Yeah.
Okay. So do we want to do these outro questions?
Well, I don't think they really apply.
Because we didn't pick the paper.
Yeah, so this was a voted paper. Very few people are voting. I don't think we'll do a vote again unless
we have a change in viewership. If you have a paper that you want us to do, email us. And we'll do it.
Yeah. That's all for this week. We'll see you again next Monday. If you're listening to this on like a Spotify or an Apple Music, I recently learned that you can rate us. So rate us 5 stars and maybe someone will discover this podcast and learn a little bit about science. If you are listening to this on Patreon, you are the only person. We only have one listener on Patreon. That is not an appreciated form of a content distribution. It seems like most people are either Apple Music, Spotify, or YouTube. If you are a scientist and you would like to do some recorded little science games on this podcast through Zoom or Skype or I don't know, something, we have some games that we want to do, some science drafts like we've been doing at home with friends that we'd like to record. Let us know if you would be interested in doing that. And that's it. Thank you for listening all the way to the end.
Woo! Bye.